In this first presentation, we're just going to review basic aspects of the immunology behind the use of GMA columns as a therapeutic strategy for treating UC patients. I'm not having any other indication, so I assume now you all can hear me and see me correctly. So I will move forward. Okay. So the immune system, as you all know, the immune system is a very complex network of cells and molecules that are constantly interacting. And in addition to all the resident immune cell populations, which are mainly focused on surveillance, another hallmark of the inflammatory response is cell recruitment. And this allows the system to deliver a coordinated and very efficient immune response. There are several factors involved or that contribute to the pathogenesis of ulcerative colitis. I'm going to go very quickly on this. In the first place, there's a decreased diversity in gut microbiota, and also it changes its metabolic profile. For instance, it reduces its ability to produce short-chain fatty acids. In second place, patients with ulcerative colitis have a thinner mucus layer with a decreased production of colonic mucin. And in third place, there's a disrupted epithelial barrier, mainly because of a reduced expression of tight junction proteins and also an increased apoptotic rate of epithelial cells. And all these factors together favor the trespassing of bacteria and bacterial products that get in contact with macrophages, with antigen-presenting cells, starting an immune response. These cells are able to produce chemokines, and these chemokines attract neutrophils. And also the monocytes that are recruited from the bloodstream, they become mature macrophages, and they start secreting pro-inflammatory cytokines which help differentiate the adaptive immune response. Among recruited cells, neutrophils and monocytes are myeloid cells with a very potent inflammatory capacity when their receptors are activated with the proper stimuli. For instance, the Fc gamma receptors and also the complement C3 fraction receptor bind immune globulins and the complement C3b fraction, and respectively. And after recognition of these ligands, they start secreting inflammatory cytokines, and also they increase their oxidative stress by favoring the production of reactive oxygen species. And all this array of cytokines and of inflammatory mediators are going to drive the continuation of the immune response towards its adaptive activity, which is characterized by the clonal expansion of specific T helper subpopulations, which are going to contribute to perpetuate inflammation in the affected areas, in the affected tissue. I wanted to stop for a couple of minutes on neutrophils because they are a very interesting population. They're kind of a double-edged sword because on the one hand, they have clear-cut beneficial effects because they are very potent phagocytic cells and help in the resolution of inflammation, mainly through activities such as apoptosis or angiogenesis. But at the same time, many of the products that these cells are able to produce, products such as elastase, myeloperoxidases, metalloproteinases, histones, are cytotoxic, and when they are produced constantly and they are liberated constantly from their nets, they generate cytotoxicity, and they enhance the structural damage that is developed in affected tissue. The liberation of net products, it's the so-called netosis, has been demonstrated in ulcerative colitis. This is just one example. You can see here how some of net products such as elastase or myeloperoxidase or histones are significantly increasing colonic mucosa from UC patients compared with negative controls. And also, for instance, the expression of PAD4, which is also a net product, is significantly increased in UC patients, and within UC patients, you can see here it's more expressed in inflamed versus non-inflamed areas. And also through this experiment, immunofluorescence experiment, you can see here how two net products such as the elastase or the myeloperoxidase colocalize in the colonic mucosa of UC patients, demonstrating the presence of these nets in samples from patients. This is another example. In this case, mononuclear cells isolated from lamina propria were put in culture, either unstimulated or stimulated with nets for 24 hours. And as you can see here, stimulation with nets induced an increment in the transcriptional expression of pro-inflammatory cytokines IL-1 beta and TNF-alpha. And this experiment was also controlled with digested nets that were not able to induce this transcriptional activation of the inflammatory response. So here you have a diagram of the granulocyte monocyte apheresis circuit, and as you can see here, activated neutrophils or monocytes expressing receptor, the Fc gamma receptors and the complement C3 receptor bind to the beads coated with immunoglobulin and with the complement C3b fraction. And with this mechanism, this strategy of the treatment aims at downregulating innate cell recruitment to inflamed tissues, downregulate inflammatory mediators, and also intends to have a further effect over the rest of inflammatory cells. We're going to see a few examples to see how it works. In this one, for instance, you can see here in blood how after treatment with GMA, we have a significant reduction in both neutrophils and monocytes. I think this is 30 UC patients after 5 sessions, I think. There's a significant reduction in both populations. Also by flow cytometry, and this is a very interesting result, we have a reduction in the population in the CD14 positive, CD16 positive monocyte population. Very quickly, CD16 is a marker of more mature phenotype of monocytes associated with proliferation, with differentiation, cell-to-cell adhesion, and so on. So the reduction after treatment in this population is suggesting a reduction in the inflammatory capacity of the monocytes that are circulating. This other study also showed a reduction in plasma levels of interleukin-8, which is one of the products released by CD16 positive monocytes, along with other pro-inflammatory cytokines. And as you can see here, they are, it is significantly downregulated 2 weeks after GMA treatment, and also the expression of TLR2, which is surface receptor for specific bacterial antigens, is also reduced after GMA treatment in patients, in UC patients. Regarding cytokines, we have some conflicting results. For instance, in this study, the use of GMA treatment was able to induce a significant increase in the amount of IL-10, of interleukin-10, which you will know it's a classical anti-inflammatory cytokine. You can see here a significant increment in this cytokine. But in the same study, a battery of different pro-inflammatory cytokines was also tested, and there was not a significant downregulation for any of them. You can see here, for instance, IL-1 beta, IL-6, interferon gamma, and so on. However, for instance, in intestinal mucosa, this other study was able to observe a reduction in the concentration of different pro-inflammatory cytokines after GMA treatment. And when patients were distributed according to these clinical outcomes, the clinical remission or endoscopic remission, they observed how this reduction in the cytokine expression, the cytokine concentration, was only present in the group of patients reaching those clinical outcomes. Regarding neutrophils, this is also a very interesting experiment that I wanted to comment because The transition of neutrophils from immature to a mature phenotype can be followed by a set of CD markers, different CD markers. For instance, immature neutrophils have are CD10 negative. However, when they become mature, they are CD10 positive. And as you can see here in the right-hand panel, a CD10 negative subpopulation of neutrophils is detectable as soon as 30 minutes after GMA treatment, and is still there 60 minutes after treatment. And this result is interesting because it suggests kind of a neutrophil renewal from very mature, very inflammatory neutrophils towards a population of more immature, less inflammatory neutrophils after treatment. And all the inflammatory milieu that normally characterizes ulcerative colitis is usually supplemented by a reduction in the tolerogenic response. You can see here, for instance, in this first panel, how the number of activated regulatory T cells is reduced in UC patients. This is detected by or they are identified as CD4 positive, FOXP3 positive, CD25 high cells. This number of cells is significantly reduced in refractory patients, in patients with refractory UC. When these patients are treated with GMA, and the ones who respond show a significant increase in this population, reaching values very similar to those present in controls. And 10 weeks after treatment, you can see here how the percentages of activated regulatory T cells present in patients and in controls, in healthy controls, are very much alike. So this would suggest restoration or a partial restoration of the tolerogenic response after GMA treatment. Well, regarding cell migration, there are not many studies evaluating the role of the GMA treatment on cell homing markers. This one, for instance, showed that the expression of L-selectin was significantly decreased after the use of beads, and in consequence, the number of PMN cells adherent to endothelial cells in culture were also significantly reduced. This is kind of indirect evidence of the effect of GMA treatment effect on cell homing markers. But, the cell adhesion molecules, integrins, and so on have an uneven expression along the colonic tissue. And I think this is kind of a research gap in terms of what's GMA treatment doing regarding the expression of homing markers. And this becomes even more important because recently, for instance, integrin alpha 4 beta 7 integrin, which is classically related to be expressed in T cells, has been described in this newly identified population of monocytes that was called non-classical monocytes. And this is very relevant because potentially GMA treatment has the power to target these non-classical monocytes, these circulating monocytes expressing cell homing markers to colonic tissue. And this would be, if it's confirmed, it would be something very, very cool. And I also would like to remind the concept of evolving immune response. This is a diagram for Crohn's disease, but I think it also is still valid for representing or showing that immune response during the progression of inflammatory diseases is different. I mean, it's not the same in early, mid, or advanced stages of inflammatory diseases. So the inflammatory cells, the inflammatory mediators that are liberated, they are changing with progression of the inflammation of the disease. And this, I think we have to keep this in mind because treatments may not have the same effect in different stages of disease and may not have the same behavior. For instance, in advanced stages of inflammatory diseases, we can already see resident memory T cells in the tissue. These cells have a very potent inflammatory capacity, and they are very fast, and the most important, they are completely independent of any myeloid cell recruitment for their activation. And this would suggest that the GMA treatment would have a very much higher effect on early stages of disease and on mild inflammatory phenotypes of disease, rather than in advanced stages where these resident memory T cells are already on site. And this was all from my side. I was just to remind you a few things here. Just remember that recruitment of myeloid cells during the inflammatory process brings neutrophils and monocytes into damaged tissue. And these cell types recruit mediators that feed back inflammation. At the same time, cytotoxic products generated during this chronicity can further contribute to tissue damage. GMA reduces activated neutrophil and monocyte count circulating in blood, and therefore it downregulates the pro-inflammatory burden into this damaged tissue. And it's also important to know that the inflammatory microenvironment may benefit from this, I don't know if depletion, but downregulation, allowing a partial restoration of the tolerogenic activity, which is very much important for the counterbalancing of the inflammatory response. And also remember that dynamics of the immune response in IBD suggest a greater GMA effect on early stages of the inflammatory course before these resident memory T cells can appear in the damaged tissue. And that was all on my side. Thank you very much for your attention.